Yeah, so for the last talk of the day, I think you heard today from so many of different domains how AI is being applied. Here we'll now hear something fundamental in terms of how AI can enable learning mechanisms, especially in a causal manner. And we have Caroline Uhler from MIT, who's full professor there, and she's at the both electrical engineering and computer science, as well as the Institute of Data Systems and Society. And she's also a core institute member at the Broad, as well as co-directs the Eric and Wendy Schmidt Center. And she's gotten her degrees from University of Zurich, as well as from UC Berkeley, and spent 3 years as assistant professor at IST Austria before joining MIT at 2015. She's a Simons Investigator, Sloan Research Fellow, and an elected member of the International Statistical Institute. And she's received a number of awards, such as the NIH New Innovator Award, NSF Career Award, the Sofia Kowalska, something that I can't pronounce very well, from the Humboldt Foundation, and START Award from Austrian Science Foundation. And here she'll be talking about causal inference.
Thank you very much, Anima. Okay, so thank you very much for having me here. I'm very excited to be here. So today we'll talk a little bit about causality and how I'm thinking about the field. And then in particular, I work on applications in the biomedical field. So in particular, telling you a little bit about how that motivates my thinking about causality. And I want to start with this one slide here, which, relates very nicely to one of the talks that we have seen in the morning. And, some of the unique opportunities. So why, the biomedical sciences and AI has to come together. Well, first of all, and we've already seen many of these talks, of course, we have all these like really amazing data sets coming out of the biomedical field, and there's just no other way of making sense of them than actually using some machine learning methods. But I want to in particular point to this plot here, which just tells you for one lab at the Broad, so just the sequencing platform at the Broad, so not taking any imaging data into account, the amount of data that was produced this year. So we heard this morning something about 10 petabytes. So this is one lab, just last year produced 80 petabytes of data. And as I said, so no imaging data is even taken into account here. And in fact, now I have to go based on what I can find online, but if you look online, this curve basically, and where we're currently, so these 80 petabytes of data, that's very similar to how much Twitter has produced in terms of data, and even the growth curve looks very similar. Now compare that to the whole Netflix movie corpus, which is only 60 petabytes of data. And now, of course, if you're thinking about what all this sequencing data is, right, this is like, has also huge privacy concerns, et cetera, et cetera. And, this kind of data is in the cloud. And so I think, people here have thought very hard about how to make this kind of data sets accessible. And so I think there is a lot that one could learn from each other. So that's the whole motivation for actually bringing these different fields together. But at the same time, I also think it is very important to, and it has been very nicely emphasized in Jen's talk just before, to always be aware about what was actually the main motivators of the methods in machine learning that have driven all of the foundational developments in machine learning. And, you were talking mainly about language models. But before that, the foundations of machine learning were things like recommender systems or online advertising or self-driving cars, right? So these were really the motivators for all of the foundational developments that have come in machine learning. Now, in these types of applications, just optimizing prediction accuracy has often been sufficient. But now here I have the example of the biomedical field, but I think, even, if we're thinking this morning, right, when it was said like, well, I'm a scientist, what it means is I want to get at mechanisms. And so also in the biomedical field, the ultimate goal is, sometimes to optimize prediction accuracy, but generally the ultimate goal is to get at the underlying mechanisms. And so here, for example, in biology, we would like to know, well, we've also seen this morning, for example, how genes interact. And there you would have some underlying causal mechanisms of what happens when I'm perturbing the system, right? I would like to be able to predict what happens there. Or in these climate models, how an intervention in the system will actually affect the long-term outcome. So the ultimate goals are very, very different. And so then that, of course, then begs the question whether or not the methods also have to be quite different, right? And so here, a prediction, as we know, is just simply not the same as something about the underlying causal mechanism. I mean, think of like a recommender system. There, it's sufficient to know that, if like the consumption of ice cream is high, probably also, people are interested in buying sunscreen. But you don't really care about why that's the case, right? If you're just, say, Amazon and need to make a lot of money with it. You really don't care about the underlying mechanism. However, if you actually do care about the biological field, and there the ultimate goal is to understand these underlying causal arrows that I have here. Now, the question is, of course, we all know correlation is not causation, and what are the kinds of methods that you can use in order to get at causality? And so here in the biomedical field, we have access to data sets that basically no other field has access to. Namely, we have the ability to perform very large-scale perturbations. And, this was talked about in Anshul's talk this morning, right? You can actually go in and you can, change the genome, or you can, make big changes of like just taking out a whole gene and seeing what actually happens when you do these types of perturbations. Now, why does that allow you to get a causality? Well, think of this particular example here when you want to know whether, like good weather increases ice cream consumption or whether it is the other way around, right? You take a gloomy day and you go out and you give out a whole lot of free ice cream and you see that that doesn't change the weather. Right? That's what it means to do an intervention and how that can actually tell you what is upstream and what is downstream. And so that's kind of the hammer that we have in the biomedical field, which you really don't have in many other fields. And that also means that, the types of theoretical frameworks who are actually using this type of data in order to get at causality has not really been developed. Okay, and that kind of motivates you that, if you have different questions and you have different tools, and of course, that also has the potential to inspire profound new avenues of machine learning research that really have to be done in order to make the most out of, out of these kinds of biological questions that we have here. And so, as was said, I co-direct this Eric and Wendy Schmidt Center, and for us, the goal is really to go both ways, right? So there is one way, and I think also from the talks this morning, I think there is a lot that can be done by using machine learning methods that have been developed and just bringing them into the biomedical field or bringing them into climate science. But I think what is equally important is actually that we change what machine learning is today, just always being aware that, the foundations that have been developed with these types of applications in mind. And so that's really what my research has been a lot about, is trying to push, the machine learning research on the foundational level through biomedical applications. Okay, so here is the types of perturbational data that I'm thinking of, and where we organize a challenge in order to make people aware in machine learning of, these other types of questions that come up from the biomedical field. I think at some point this morning, or I think it was just mentioned, there was something about immunotherapy. And, so our immune system is capable of killing cancer cells. And so what immunotherapy, but the problem is that cancer cells send out signals that make these immune cells, exhausted, and then they don't fight them anymore. So in immunotherapy, what you want to do is to make changes to your own immune system, to your own immune cells, so that they are very effective at killing cancer cells. Okay, so the question is, can we find the right changes to these immune cells in order to make them be very effective at killing cancer cells, so that your own immune system can actually do this? And so these are like the types of questions that then come up. Like, we have like, here we're just looking at, so humans have, was said this morning, about 20,000 genes. So here we're just looking at perturbations at every gene. Okay, but you could actually make combinatorial perturbations. But in this particular challenge, we're just looking at, you could make a perturbation in every gene. And now here you have to test these out in the mouse. So in this particular experiment, all we were able to do is 70 different perturbations. Okay, so we chose 70 genes, which we perturbed. And then what you get out is, you get many, many T cells, these immune cells, these, these are the soldiers in your immune system that are the ones that are effective at killing cancer cells. And you see in which state they are. Okay, so here we just divided all of the cells into 5 different states. We don't want our T cells to be here. We really want them to be in these kinds of states over here, right? In the effector and in the progenitor state. Cycling are just the ones that are currently dividing, and then we have some other state which we'll not care about here. And so now how this data looks like is, if I don't make any change, then that's how the picture looks like, how the distribution of T cells is. And then now you have 70 of these types of pictures where you see, hey, if I make that change in gene I, then this is how the picture looks like. I have quite a lot of cells here, but I don't really have as many here. Right? And so the question is, can you now come up out of these, I give you 70, can you now predict out of all the 20,000 that you could possibly do, which ones should I do in order to move the cells to a particular state that I care about? So it's this design question, and it's a huge combinatorial space, and it's in fact even continuous, so it is really infinite because, you could even change dosage and things like this. And it's a causal question, as I said, right? If I do an intervention, only things downstream change. So it's not conditioning. It is a causal question. It's like the ice cream and weather example, right? If I change the ice cream consumption, everything upstream is not going to change. So it is really not your standard prediction task because I am trying to predict the effect of an intervention in the system. Okay, so these are the types of data sets that we can get. And, it's also a really nice setting where, yes, the questions are different because we can perform these interventions, but also it's a nice way of testing how well your causal inference algorithms work. Okay, so how do we generally think about causality? So, this is now again from biology, but, generally, when we're thinking about causal systems, and generally we think about these directed graphs, these directed networks. These are the types of, directed networks that people have built with, a huge amount of biological pre-knowledge and a huge amount of experiments. But generally, the statistical theory for causality and for thinking about these causal networks was developed by Sewell Wright, a geneticist, in fact, where, every one of these edges directed edges is a causal link. And how you should read these networks is, every one of these nodes, say every one of them is a gene, and this is how much this gene is expressed. Go forward. Every one of these is a gene. Say we have 20,000 of these. And so how you should read these kinds of networks, and this is the theory that was developed by Sewell Wright, is every node is just some function of its direct parents and noise. Okay, so here, for example, X4 is some function of X2 and X3 and then some noise. And again, these functions don't have to be linear, of course, they are generally not. And the noise doesn't have to be Gaussian, it is generally not, right? So this is really the standard way of representing it. If you know Bayesian networks, then maybe you also see these kinds of things, right? But this is actually a causal model. The difference is that it also encodes what happens when you're doing an intervention. Right, so it does encode. If I, for example, here do a perturbation on X2, then I'm setting X2 to some variable, well, then X1 doesn't change. Okay, so these are these causal structural equation models. And, what we're really caring about in these settings is, of course, generally I don't know the underlying causal network, right? In my T cell example before, if I would give it to you, you would know which intervention you should do in order to move cells from one state to another. You don't know that, so the whole problem is about learning these networks from data on the nodes. Now, Peter Spertus at CMU has laid out really the foundations for causal structure discovery. So what can you do from just observational data on the nodes? What can you learn? What can you not learn, etc.? Coming up with the first algorithms to do that. And we were quite excited, but this is not what I'm going to talk about. We were quite excited when all these large-scale perturbational data sets came out to think about, well, how much does perturbations actually help us? Okay, so if I also have perturbational data, then, as I already did motivational before, right, definitely that tells you something more about directions. But the question is, how much does it really help you in terms of, say, sample sizes, in terms of, maybe more identifiability, etc. And, yes, we were super excited at the beginning, but very quickly understood and have proofs. And these are kind of the guidelines that even to learn graphs of, say, very small sizes, 10 to 100 nodes, you need huge sample sizes, okay? So even for these 10 to 100 nodes with interventions on all of the different nodes, you still need sample sizes of the size like 10 to the 6 to 10 to the 7, which, if I want to learn a network on 20,000 nodes, we will never be there, okay? So even in like these very large, so we do get sample sizes of like 1 million per experiment in the biological settings, but still you'll never be able to learn a graph on 20,000 nodes. So then the question for us was, what can we do, right? If this is not really what we can do. And so this is what I want to talk about here is like two different problems where I'm thinking that maybe, you know, yes, going through learning the causal structure has always been what, you know, the kind of the fundamental goal was in causal inference. But maybe, you know, if we cannot get there, maybe there are still some other interesting problems that we can get to that might be easier than actually learning the full causal graph and then trying to predict what a particular intervention does. And so here are two problems that are very fundamental in the biomedical sciences, and I guess probably in many scientific areas. And I'll just present them separately. So one of them is the following here, which is, you know, say I have some data set for different types of perturbations. You know, these can be drug perturbations, they can be all these genetic perturbations, whatever it is. And they were measured in, say, a mouse. Okay. Now, what you care about in pharma is like, hey, I measure all these perturbations in a mouse. I would like to predict what it does in humans. Okay, so that's transporting across different contexts. Now, that's a huge transport, but let me do it in a different setting. Like I have all these perturbations that were performed, and there is, and I'll show you an example, in like a large-scale cancer data set. Now comes a new disease, which is SARS-CoV-2. Right now, I would like to predict, without having to redo this whole experiment, well, what would these same perturbations do in this new context, which is this new disease context? Or I know what it does in one cell type, I would like to know what it does in another cell type. Okay, so that's transporting to new contexts. And then you have another problem, which is transporting across different perturbations. So as I said before, right, we only did 70 different perturbations. There are large-scale perturbation screens, but you'll never be able to do all combinations. Oops. You'll never be able to do like, and I'm not just talking about pairwise combinations and genetic perturbations. You care about doing 10 perturbations at the same time or 5 perturbations at the same time. Right, so I'll never be able to do that on 20,000 nodes. So I'll have like some, and also, you know, the space of chemical perturbations is huge, of course. So, you know, you can do some perturbations, and now from these perturbations, you would like to predict what other perturbations do to these cells. Okay, so this is transporting to new types of perturbations. So these are the two problems that I'll be looking at. Also here I chose on purpose two different modalities, right? In biology, we measure many different types of modalities. We have seen a lot this morning on sequencing modalities, but you know, we much cheaper than getting sequencing is images. So these are images of single cells where I'm getting to show you is for example, one fluorescent label, which is just the nucleus. And again, you know, there are these very large scale data sets, many perturbations applied, and all you get to see is like these images of single cells. Okay, so the question is, how do I go about these problems? And first, I want to get to some of the more fundamental kinds of questions on the machine learning side. So definitely what this all is always generative questions, right? I'm trying to generate what the data looks like under a particular, in a new context or under a new perturbation. Okay, so let me just set up the framework that I'm going to use for these generative models. So we have been working a lot on autoencoders just because we have over the years gained quite a good understanding of what kinds of function classes they learn. But let me just first start with a paper that is actually coming out this week, which is, you know, just giving you so we always work with these overparameterized autoencoders. And so I'll want to give a little bit of intuition for why overparameterization. So first of all, I'm sure many here are aware of Misha Belkin's and co-authors' really very pretty work on, you know, showing that deep neural networks in the classification setting, it can generalize well, although it is overparameterized, right? So although you get down to training error 0, you would think it's overfitting, right? And maybe it cannot generalize, but we have all this work by Misha and many others that, you know, you can still generalize. Now, what is also really interesting is, of course, is this work by Jacob et al. On the neural tangent kernel. So, you know, generally, we always think in this way that if you make neural networks larger and larger, your computational costs are going up more and more. But the interesting thing is if you go to infinite width, then suddenly you can write down the function that is being learned by your neural network in closed form. And that's what this neural tangent kernel is. Okay, so suddenly your computational cost goes down in the limit as width goes to infinity. So So these two things kind of motivate you to actually look at this over-parameterized setting and maybe end the limit as actually the width goes to infinity. So what we were kind of surprised by is that, even though neural networks are used in all these very mission critical domains, nobody ever proved that they're actually Bayes optimal. Okay, that they are really the optimal classifier that you could possibly use. Not that they can approximate any function, right? But when you're training them, that they are in fact the best possible thing you could possibly do. So we took these infinitely wide network and we made them also infinitely deep. So that the only parameter you care about is now the activation function, right? I have nothing anymore that I have to worry about in the whole architecture. So I take them infinitely wide and then infinitely deep. Now all that matters is just the activation function. And so here we completely classified all classifiers that you can get out with any activation function. And in fact, you only get 3 different classes. One of them is optimal. So neural networks are Bayes optimal, and we completely classify all activation functions that are Bayes optimal. All the only other classes are it either does majority vote or nearest neighbor, which are not optimal. And then there are in here are the optimal ones. What is interesting is none of the standard activation functions are optimal. So ReLU, sigmoid, anything that anyone has ever used is not optimal. But you have very simple activation functions that are optimal, and you can actually use them, and we have used them. So they're just some degree 3 polynomials and others, and you will see in applications that they actually do better. So this is all in the classification setting. Now, in the biomedical field, we generally use the representation learning setting. So the question is, what can we take from here? And this, of course, motivated us to look at over-parameterization also in the autoencoder setting. Or, I mean, autoencoder is the only thing we can do theory on. Once you have these kinds of things for GANs, and I'm happy to use them. But until I have no understanding really of what the functions are that are being learned, I prefer to use simpler models. So autoencoders are, 2 neural networks here. We're going from an input space to this latent space representation and then back to the input space. Now, what does it mean to be over-parameterized? I'm going to make instead of using this for dimension reduction, I'm actually going to have the latent space be larger dimension. Okay, now this may sound crazy again, right? So definitely I'm getting 0 training error. I have enough parameters. I have more parameters than I have samples. So I'll always get 0 training error. And I could always just learn the identity map, right? Which would be crazy because then why am I even doing this? But I'll show you that we're not learning the identity map and we have proofs, okay? So, and you can very easily see that you're not learning the identity map. So you just set up some autoencoder like this and you just train it, okay? So if it was the identity map, I could put in any test example and out should come the test example. And generally, something comes out that looks like the test example if you've done these autoencoders. But, if it was a test example, then I could put it back in and it shouldn't move, right? If it was the identity map. Well, so just do that, okay? And you do that many times and, you can put in a training example with some noise on top. And if you already iterate it many times, it doesn't look quite the same anymore. And you continue, and this is a proof that you will always end up in one of your training examples. Okay, so the maps that are being learned by these over-parameterized autoencoders, yes, they could learn the identity map, but they don't. They're hugely self-regularized, and this just comes from gradient, right? Gradient descent could go to any one of these infinitely many solutions that have 0 training error, but what it goes to is a solution that is highly contractive at the training example. Okay, so these are always our neural networks that we're using are always in this regime where we're doing something that might seem crazy, where we're like hugely over-parameterizing them. We're using autoencoders to embed into a higher dimensional space than what we're starting off. Okay, so let me show you, and these are again the problems that I want to work on, but these are again the autoencoders that I'm going to use. Okay? Now this all has nothing to do with causality yet. It's very dry. Okay, so this all has nothing to do with causality yet, and now I want to bring in one more point, which is why I added in here these images. So, causality or causal inference always starts with one main assumption, which is what I showed you before, that you're given nodes and you want to learn directed edges. right? I want to learn my causal links. Well, now here I have these wonderful data sets, so much cheaper than everything over there, right? Huge, I have large scale perturbations. You also have it in any of the climate questions. You also have a whole lot of imaging data. Well, what are my causal nodes? Right? Certainly not pixels, because pixel 1 in this image has nothing to do with pixel 1 here, has nothing to do with pixel 1 here, right? Because I have no reference system. So is it, the shape of this thing? Is it, how much protein I have inside the cell nucleus? Is it where it is localized, etc.? Any of these things could be causal variables, right? But I'm not giving you the causal variables. So this assumption is not there when you're working with imaging data. You actually don't know what the causal variables are. And I would argue even in sequencing space, well, it's just because we set up how we're measuring it that we have this reference system of genes. And so we're always just drawing it as these 20,000 nodes, or, if you do it in proteins, okay, you'll have more, right? But that might not be the causal units that we actually care about. It might be some groups of genes that are working together, etc. So just because this is how we're measuring it, and it's very clear in the imaging space, right? Nobody tells us what the causal variables are. So the question is, how do we even learn causal variables? Okay, so that's what I want to talk about. And this is where our autoencoders will come in. So here I'm proposing that maybe representation learning can actually be used as a tool for causal feature discovery. So causal features is what I mean by causal nodes or causal variables. Don't really know what kind of name we should use for this. And so here what we're using, and I will have some initial proofs on one of the next slides, that here in this case, I'm proposing to do this by using the fact that in the biomedical field, we often have multiple modalities. Okay, so here we do have single cell imaging data, and we do have all kinds of other single cell sequencing data sets. And here the idea is like very, very simple, and then you have to make it kind of theoretically grounded. But, we have a system which are these cells, right? So, I have cells of a particular cell type, say, and I can look at them from different views, right? Like I can look at them from imaging land, or I can look at them from sequencing land, or I can look at them from any other land. So if I care about what is causal in them, it should be there in any kind of view I'm taking to them, right? Whether I'm looking at them from the left or from the right, whatever is causal should still be there. And so that's all we're doing here. So we're trying to learn representations that are invariant to how you're looking at the system. And I'm proposing that, these causal features, so whatever is still invariant to how you're looking at it is more likely to be causal than something that is only there in one of the modalities. So you can kind of get rid of a whole lot of spurious features or spurious correlations by doing this. Now, how do you do this with autoencoders? Even if you don't have paired data, but you know that at random, you took out some for imaging, and at random, you took out some for sequencing, well, then in the latent space, the two distributions have to match. Right, because it's the same cells. I just at random took out some for sequencing and some for imaging. And so you can just have another loss function in the latent space to make sure that these two distributions match. And then in this way, you can also translate and you have a better representation there, which we propose here. And I will show you have initial proofs that this is in fact more causal than what you would have in each one of the different modalities. But first, just to show you why over-parameterized, so what we use is over-parameterized autoencoders. Just to show you with images, I won't go through proofs of why this should be more causal and should really be the right thing to do. Here I'm showing you an example of nowadays, I think it was also brought up, nowadays, we can have the sequencing data, not just for single cells, but actually inside the tissue. So we get to see where the cells are and we get sequencing data. Now here, just to go the other way around, to make the climate people laugh about where we are in single cell biology space. So the standard way of how people are still analyzing this kind of data, which is inside a tissue, is you took your cells out of the system, okay? So I take all of my pixels here out, all my cells, and I do clustering on them. Okay, so it's like you have your image and you dissociate it, and you take each one of them, and now you just cluster them, and then you put them back into space. And then you do your analysis back in space, okay? So I'm sure that nobody who does anything in physical land or in climate would ever do something like this where they're doing the analysis without their 3D environment present throughout. So that's still the standard, just because, you know, many people are coming from the single cell biology land where these are the methods that have been developed. But of course, so you can add in, right, the 3D context into it. And so here what you're seeing is that we're using our imaging data and our sequencing data, and you can do these, you know, multimodal, whatever, these latent spaces that have all of this different information in it. But here's just to show what happens. This is on mice and brains. This is just to show when you use your standard autoencoder versus, you know, an over-parameterized autoencoder. This is the brain, so 4 different mice slices. You see that, you know, the same regions are clustered together. Whereas, you know, if you're using any of your standard autoencoders and you just cluster your data in the latent space and you map it back to where the cells are, you see that, you know, different regions are being clustered together, which is obviously wrong, right? You don't need to know biology to know that this is certainly not a good, not good clusters that came out of your latent space. Okay, so these are the kinds of things where you can directly get it out when you're actually just using over-parameterization. You can also do things which are maybe closer, where, you know, let's take ECG data and MRI data from the heart. And, you know, again, these are just 2 representations of the heart. So again, you can do the same thing when where you're integrating it into a joint latent space. And what is quite interesting also already gives you a little bit of motivation or a little bit of a hint that maybe it is more causal than if you would do it in each one of the different representations separately. You can now do these genome-wide association studies, kind of like figuring out which variant inside in your DNA is actually associated with particular phenotypes or particular diseases. You can actually just do that now in the joint latent space. Okay, I can figure out which variants actually do something to the heart, which I would see in both of these different representations. In particular, you can do genome-wide association studies completely unsupervised. I can just look at which variants push me in any direction, meaning it does something to the heart, and I can then decode and see what these actually might be doing. Okay, but just, so what I really want to get to is just a little bit of theory of where we are in terms of thinking about this. Okay, so this is how we're thinking about it, right? We have like all these different modalities, and you're measuring like images or sequences or whatever you have. Okay, so these are these X's, these are my different modalities. And how I'm thinking about it is there is some latent network, right? My nodes, I don't even know what they are. I don't know what the causal features are. I don't know what the causal network is. And I also have some other causal variables that are domain specific. Right, I have these Z7, Z6, Z4, whatever. Those are all domain-specific ones. And I'm asking whether it is really true that this intuition that we had, that if you have multiple modalities, that should actually help you to identify what the causal variables are. And you can really prove that. Okay, so again, simplified setting for now. We only have proofs in the linear setting, linear non-Gaussian setting. But you can prove that just from having multiple modalities. What you couldn't do otherwise is, you know, with one modality, you cannot figure out how many latent variables you have, what your causal variables are. But by having multiple modalities, you can actually figure out how many causal variables you have. Also, you can figure out what actually this underlying latent causal graph is. Okay, so we're doing everything latent, right? Our nodes are latent, and, you know, we're trying to figure out what this causal structure is among these latent nodes. So this just tells you that, yeah, at least, you know, at a very simple level, it is true that multiple modalities do help you to get a causal feature discovery, and you can get very similar kinds of results when you have interventions. Okay, so somehow being invariant to different interventions is very similar to being invariant to different modalities. That's at least how we're thinking about this problem of getting at different how can we learn, you know, a structure among latent or latent causal structure from images, say, for example, what are even the causal nodes? And I think I started at 1/4 past. Is that correct? Whoever, yes, okay, sounds good. Okay, so I'm still not there yet, right? So I need to have like one more slide where I'm kind of finishing up the story. But like, you know, here, what I showed until now is, okay, from images, at least if we have multiple modalities, then we can combine multiple modalities using these over-parameterized autoencoders in order to actually figure out what are even the causal variables in this system. Okay, now how do I go about these problems, right? So in the end, I still want to go about these transport problems. I want to here, for example, be able to transport from, old context, say one disease to another new disease, or I want to be able to transport across different perturbations. Okay, so this is just, one idea, which I'll end up with. So again, I talked to you about these neural networks and we make them infinitely wide, right? And then we suddenly don't have to pay the price of computation anymore because we can compute them in closed form. Okay, so this problem of, like, if I go back here, right, this problem of going between different contexts or going between different perturbations, I could just see it as a matrix completion or tensor completion task, I guess, and this is actually a data set, if you want to play around with this, is freely available, where you have many different perturbations, drugs, genetic knockouts, whatever, and many different contexts. These are all different cancer contexts. Okay, like lung cancer, skin cancer, all kinds of different cancers. And so every dot here means that this particular data set was obtained. So meaning this particular perturbation was applied to this particular, disease context. And in fact, what you're measuring is a 1,000 dimensional vector, okay, some gene expression vector. So it is really a tensor. Now, as you see, so what would happen if a new disease context like COVID-19 comes in? Well, all I get to see is the normal state without any perturbation. So what happens when you do your standard matrix completion task? Any matrix completion algorithm will always have somewhere in the back end it has some kind of low rank. Okay, so what happens when you do low rank in these systems? Well, you fill in a constant. Right, it's a pretty bad prediction for any of these things. Okay, so what can you do? Also, it's not causal at all, right? Because anything like matrix completion is always symmetric. Whereas these are, as we said, these are perturbations. Right? If you're downstream, you only change something that is below you, whereas these things are conditionings. So, just probabilistically, conditioning on a variable is very different than intervening on it. So certainly we need some kinds of matrix completion approaches that are causal that will not be symmetric in both directions. So low rank will definitely not work, but it's also clear here why it doesn't work. Okay, so the simplest thing we can just think of, like based on everything I said until now, well, let's try to see what it means to do these infinitely wide neural networks, which we can just write down in closed form, for matrix completion. Okay, and then now you have to crank, you have to go through like whatever the math is, and you'll figure out, and I won't go into what exactly it is. You can look at this paper or ask me, but this is all we did, okay? So we just actually wrote out what it means to have an infinitely wide neural network that just figures out this matrix completion task. What is the function that is being learned? And then you just apply it. And this just straight out will beat any of these approaches that have been developed just particularly for this particular task. Okay, so all we're doing is just using these figuring out what the neural tangent kernel is for matrix completion. And in fact, you will just, directly be stronger than any of these that have ever been developed. And you're very fast. As I said, you're not training anything. You actually get it just in closed form. And you can apply it for any other question as well. Any other problem of like image and painting or whatever you want. And if you write it out, what this neural tangent kernel does, you actually have something that is kind of like a prior that you can plug in, which, generally in your neural network would just be the identity matrix. But what this prior is, what you can encode is like how similar are two cell types or how similar are two drugs to each other. And so this is a neural tangent kernel is a kernel, and you can write out that what it is a function of the inner product of whatever your priors are that you're giving. Okay, so that's how it is encoding kind of similarities between drugs or similarities between cell types that you can put in as whatever. So you can, for example, take any of these algorithms as your priors and then make them better just by running them through this particular very fast type of method. Okay, but I will want to end with that. You can then actually apply it and try to predict what all these drugs do in the context of COVID-19 is what we did here. And then, apply your causal learning algorithms to actually really figure out what are the causal proteins in this particular setting. And this one is, in fact, one that is in clinical trials, which we didn't know. Someone at the Broad told me that, in fact, there is a clinical trial ongoing for this particular one. So, but I want to end with that. I think I'm out of time. So hopefully I was able to show you that, we do have to think a little bit, I feel, more about, not just using whatever is standard in ML and just like bringing it to whichever field we're working in, but there is really a lot that you can gain out of going the other direction. out of thinking carefully about what is actually the question, what are actually the data sets, and maybe we do need to develop new theoretical and algorithmic frameworks. And by doing so, you may be able to answer very fundamental questions about machine learning. In this case, about, what are autoencoders learning? Or, should we think about causal inference in the settings where we're actually not given the causal variables themselves? I'm very excited about overparameterization. It has all these very, very nice properties about self-regularization, et cetera. And the inductive biases are just very well aligned with many of the kinds of more causal questions that we're asking. And then of course we get to the second question, which was more like Jen's view here, which we're also very excited about design, right? That once you can predict the effect of any perturbation, well, then you can also design them in order to move to a particular state. But it is a completely other question, which then leads to new types of optimization questions as well. So I'll leave you with that and just, of course, the acknowledgement. So most of the work that I presented today was done by two amazing PhD students, Adit Radhakrishnan and Chandler Squires, and then would not have been possible without amazing collaborators. Misha Belkin for the things on overparameterization, Matthias Durton, what I showed you on the latent causal variables, and then everything on the biological side was in collaboration with Shiva's lab, and then of course funding. And thank you all for your attention.
This is really awesome and inspiring. I was thinking about what is it that is assumed about the difference between different modes and why those two modes are useful. Because I mean, in theory, you could split a data set and call two separate parts of it different modes, and how would the thing know the difference, right? So what is the sort of assumption there that's making those useful in some way?
You need the assumption that there are two different distributions. Okay. Otherwise, this doesn't work. So in the theorem that will come out. But yeah, I mean, all that you're looking for is what is invariant. That's really the whole intuition behind it. And yeah, so whatever, if two things are just correlating together in one data modality and it's like just given by some latent confounder, right, you're going to get rid of it because that will be messed up. And you can even do this for different batches. I'm not saying it has to be two different modalities. It just has to be two different distributions that you're putting together. So that's why everything also that I showed you from the different mice, right, that helps you to get it all into the same system.
Yeah, that's great.
So, and in fact, this has somehow been like always. So why vision tasks work so well is because they are based on looking at what is varying together, because everyone here in the room is taking pictures and putting it online. Right? Whereas when we're taking like whatever, any kind of microscopy images, we're always trying to standardize. I don't think that's the right way to go in order to get good embeddings.
I have one other really quick question, which is if you can give intuition for why overparameterization is really useful, not in the matrix completion, but in sort of general in this. It seems like there should be an inherent lower dimensional space. That's how I'm used to thinking about it, but I must be wrong there. So.
Yeah, I mean, it is, yeah. Okay, so the question is, what does it do with the additional dimensions that it has, right? And so it does stretch certain dimensions. And so whenever these dimensions where it stretches is aligned with the biological variation, then you're doing well. If the dimensions where you're stretching is aligned with the batches, then you're going to do poorly. And so you can figure that out even when you're just looking at PCA space beforehand. And so if, at a very high level, if like in the PCA space, your largest variations are aligned with biological variation, like different cell types, this will work. If not, right, if your batch effects are the biggest variation, then this will not work. Then it will make these bigger. And we can talk more about it and why it works for causality, et cetera. We can talk more.
Thanks for a great talk, Caroline. Two questions. One is sort of a follow-up to the first question you just had about what does it mean to have these different modalities, and it reminded me a little bit of this super old technique, which I'm curious if you heard of co-training back in the day, where you say to the extent I have two views that are independent, then I can improve my model. And I'm not familiar with the theory, but I'm curious, is it connected to your theoretical? Might your theory help explain that this is indeed producing more causal models or something like this?
I think that's what it's showing. Yeah, because I think, this has always been used, right? I mean, like computer vision, like, always like language models plus images are being co-embedded. So it's not a new thing to do this. I think what we wanted to prove, so our intuition was always that it should give us something more causal. And I think we were excited that we can actually prove it. So yes, it should give more motivation for anyone who has, done this for a long time.
And then I had a separate question, which is for the autoencoder, and it's not learning the identity. Are there any particular assumptions, like how you initialize or the gradient step, like anything about the optimization, anything at all?
Yes. Okay. So there is one thing about what are the proofs, right? And then how do we just implement it? So in the proof, you have like, yes, you're initializing as close as possible to 0, which is generally what you do for all your weights. And we do gradient descent. That's all we're able to prove. Now, when you're running it, it's actually quite exciting. So we have run it with many different. So in all of them, you will at some point see this. Okay, you just need to be for some kinds of optimization methods or for some kinds of activation functions, you need to be more or less over-parameterized to see this. But in all of them, you will see the behavior. We don't have proofs because, why we will never end because everyone cares about some other method of doing it. Yeah. So, and the nice thing is also once you are enough, so how do we even figure out what over-parameterized means? Well, at some point it stabilizes, which is also great. So just make it large enough and you'll see that, no, this is also great, right? Because otherwise you always care about like, oh, which architecture, etc. But here it actually stabilizes. So we just, at some point nothing changes anymore. And then you know that you're in the right regime.
Great talk. So I was wondering about the shared latent space between multiple modalities. How do you make sure that the shared latent space doesn't over amplify common features and disregard, modality specific features?
Yeah. So as I showed you in the proof, right, this already motivates how you should do it. So you should have some of the latent. We didn't do it in the other applications, but you can, and we are now always running it like this. You just have partially shared, right? Okay. You have some, But maybe I'll actually, one more thing that I always, is maybe interesting for the people who have smaller data sets. What this also says, this over-parameterization, is that at some point you should throw away data, right? Because if you don't have enough computational resources to be over-parameterized, it is better for you to throw away data. Just as this is the consequence of this work. And we see that. Right, you definitely, so often, if you have too big data, so the data sets can be nice and they can all lie on a manifold. This over-parameterization just means that you have to be higher parameterized than that. But I think in many of the climate settings, I don't know how it is, but you may not be able to have your models be super, super over-parameterized. Just a question. I don't know what the answer is.
Okay, so one question. Could you put more color on the over-parameterized method, because one of the things I'm not clear, like if you mentioned like it's indefinitely wide. Now, when you implement that in your computation, how do you do that? Is that you replace that by a function, or it's basically how do you implement that? Yeah.
Correct. Yeah. So first of all, you can show that these things actually hold by making your classifiers wider and wider, and you see that. This was not out. So Jaco these are the people who prove that if you make your neural networks wider and wider, they will converge to this function. So when we are implementing it, we literally take the neural tangent kernel. So this is a function that you can write down. And that's why, for example, for matrix completion, they didn't write down what the function is. So our whole work was figuring out what is the neural tangent kernel? What is this function in the question of matrix completion?
So basically you need to figure out that kernel function first.
Correct. And that's math,
Okay.
And so that what goes in there. So there is a whole lot of papers. So first they did it in the fully connected setting. Then now there is a convolutional neural tangent kernel. Then you have like all kinds of different activation functions. For all of them, you have to figure it out. But I am convinced that once we have skip connections and all these things will lead to a different type of function, which you first have to mathematically figure out. I'm pretty sure that once that is all done, then you're like, this will be much better than everything.
So there's an established method to figure out that kernel function.
No, you have to do it for every single trick separately.
Oh, okay.
Yes.
All right. Thank you.
That's where the math goes in. Yeah. But many have already been done. Yeah. Time out. Yay. Okay, let's all go for wine or, but thank you all very much.